From Microbe TV, this is TWiP, This Week in Parasitism, episode 236, recorded on the 17th of May, 2024. I'm Christina Naula, and you're listening to the podcast about parasitism. If you like what we do, consider supporting the science show of Microbe TV at microbetv.com/contribute. I'd like to introduce Dixon Despommier today. Hello, Dixon. How's the
Hello,
weather like
Christina.
where you are?
Let's see. It's a little bit cloudy. The weather is tolerable. It's in the 70s. It rained a little bit this morning. It's a little muggy, but it's typical spring weather.
Very good. And I would also like to introduce a guest today. We have Marion Avril today joining us, Marion is the principal scientist and program director at MalarVx Incorporated. Welcome, Marion.
Thank you.
Hello,
Thank you for
Marion.
having me. Pleasure to be here.
Oh,
It's
yeah.
good to see you. What's the weather like where you are?
So I'm in Seattle, Washington. It's partly cloudy, partly sunny today. It's about 14 Celsius, which is 57, 58 Fahrenheit, about.
Right.
Typical spring weather.
Got it.
Typical spring weather. I can beat that. We have proper summer weather here in Glasgow, which is
Wow.
very unusual.
Wow.
I also experienced the weather in London today because I was in London yesterday, traveling home today.
Huh.
And I can say the UK is hot.
Hot. Interesting.
So, Marion, you are here today, and we're going to be talking about a paper that your
Yes.
group has presented. But let's start with talking a bit about you.
Yeah,
So,
sure.
just introduce yourself. Tell us a little bit about you, where you come from, how you became a scientist, and how you became a parasitologist. Anything you like.
All right, sounds great. So I'm Marion Avril. I came from France in 2005. I came to Seattle area for my postdoc. So back in France, I did my PhD in infectious disease under the direction of Jure Geisen. And I was located in Marseille at the University of Medicine in Marseille. And I was already studying malaria, actually. I was focusing on pregnancy-associated malaria for my PhD. And I had the opportunity to join the lab of Dr. Joe Smith here in Seattle for my postdoc. So I came to Seattle at the end of 2005. And I did 5 years postdoc, and then I continue on 5 years staff scientist and senior scientist in the same lab at Seattle Biomedical Research Institute, who became Seattle Biomed, and toward the end, before I left, I became Seattle Children in Seattle. So I study malaria pretty much all my career life so far, except a short window where I did at the University of Washington for 9 months. I studied microbiome between the job at Seattle Children and the job at MalarVx. I went to the University of Washington, and I should say it was a fantastic eye-opener. The microbiome world was fascinating. But unfortunately, because of funding situation, I had to look for a new opportunity, a new job, and I had the opportunity to join MalarVx in June 2020. And since now, I've been working with my team at MalarVx, and our CEO, it's James David, since he created the company. So MalarVx is a company that the paper we're going to talk today, it's looking at the replicating RNA-based vaccine against malaria. But MalarVx was originally created to create a in vitro sporozoite production as well. So we have two topics of science in the lab. We're working on producing the different stage of the mosquito stage in vitro, and we're doing the repRNA based approach. So today I'm going to talk about the paper that was published recently in January in NPG Vaccine. And yes.
That sounds really good. Just before we dive into the paper,
Yep.
I thought I'd kind of maybe wanted to learn a little bit more about you. And I fell into science by accident. So a lot of our guests say, or on other shows they say, oh, I started to talk and walk, and then I knew I
Mhm.
wanted to be a scientist. So how was that for you?
So actually, I've always been very interested in biology. As a little kid, I had unfortunately I had a few little health issue as a kid. I was born highly premature, and then at the age of 9, I was diagnosed with a myasthenia gravis, and that
Oh,
was a very difficult time. But I had surgery, and which saved my life, I would say. At the age of 4, I also went having a very bad heat stroke with potential neurological issue. So I would say, as a little kids, I don't think you realize, the biology behind. But as I grew up and went to high school and get into the science, the biology, I found that fascinating, and I wanted to learn more about the biology in the human being, and to know and understand a little more, for example, the immunology and all that stuff. So that was my passion for science. I think grow over time with years, and I think I was just very curious to understand what the disease and how the human body, fight the different disease. And I knew when I was at high school, I was interested with med school, I would say,
Mhm.
but I wanted to be a surgeon. And I said, that's cool, but maybe I want to do more of that immunology and understand more disease related, human disease related. So it's why I went to the University of Biology and study biology, and then immunology and infectious disease, and happened to have the opportunity to join lab working on malaria. So it's how I started. It was just opportunity one after the other. And here I am today and working on a small biotech, working on malaria again, and I love it.
And you've never looked back since. That's
Yeah,
amazing. That's a really,
yeah.
really good story. Okay, so shall we
Sure,
talk about your paper?
sure,
I'm really
yeah.
excited
absolutely.
to talk about it. It's a really good paper. I enjoyed
Thank
reading
you.
it. I read it on the train from London to Glasgow, and usually these train trips are really, really boring because the Wi-Fi is not working, and the coffee shop is closed. So I had really excellent entertainment today.
Great.
So
Thank
yeah.
you. So I would say, first of all, it was a great collaborative work. Okay. MalarVx was not the only player in that paper. We have a great collaboration with the team at HDT Bio. It's another company here in Seattle. The CEO is Steve Reed, and Jesse Erasmus and Annemiek Kender are both director of virology and formulation, respectively, in that company. We also work with Dr. Sean Murphy and Melanie Shears and Felicia Watson at the University of Washington here in Seattle. So my team, Zach and Kiara MalarVx, and I started this repRNA-based vaccine about 4 years ago. Actually, that was a project that, following a discussion with a replicant self-amplifying repRNA-based vaccine that HDT Bio was developing for other infectious disease. And obviously, with the circumstance of the pandemic, they were focusing on COVID, but they had very good result with their COVID vaccine repRNA. So we said, okay, can we look into a repRNA-based vaccine for malaria? It sounds very promising. And so what we took the approach, it was to first look into what a prime boost uh, uh, regimen of immunization would do as a repRNA only based vaccine for malaria. what would be? So I would say the innovation in our vaccine approach has multiple little components. First, the replicant RNA, it's fairly new, I would say, in the world of malaria approach. Also, we combining the replicant RNA with a formulation which has been developed by HDT Bio. It's called Lion. It's a lipid inorganic nanoparticle, which is different from an LNP. And as you may see in figure 1, the big difference with an LNP, the repRNA, the RNA, instead of being encapsulated, it's around the lion. So the RNA, it's not inside the lipid nanoparticle.
Oh, okay, so it's stuck around it.
It's
Yeah.
stuck around
Okay.
it. So the
Yeah.
lion protect the RNA from the outside.
Okay. Can I just ask before we
Yeah.
dive into the details of the paper, because I've got a million and one questions that I've noted down.
Yeah.
So, just a couple of very, I don't know, very basic questions probably.
Yeah.
So I know you have done these experiments in mice, and you've
Yeah.
been using a parasite strain, Plasmodium uelei, and
Yeah.
I was wondering maybe if you could tell us a little bit more about the choice. I mean, obviously, it makes sense to use mice, but maybe tell us a little bit also about that particular parasite because I'm not particularly familiar with it, and I'm
Yeah.
not sure if our readers are. So, yeah.
Yeah. So do you want to talk about the biology of the parasite itself in
Well,
general?
maybe just more kind of what. eh, eh, To what extent or how does it differ from Plasmodium falciparum, maybe?
Mm-hmm.
And, is it easy to work with in the lab?
Okay.
You talked
Yes.
about culturing, so we can maybe later talking about the sporocyte culturing as well.
Right.
So yeah,
Right.
just so that we know
Yes.
why it's a good model for Plasmodium falciparum.
Absolutely. So the Plasmodium uelei, the strain that we use here, it's because it's a rodent malaria parasite. So it can infect the mice model, and we use mostly bulb C mice in our model here. So the Plasmodium uelei express different protein on the surface, and the protein that we choose in this study is called the circumsporozoite protein, CSP, and it's protein immunodominant protein, and it's a vaccine candidate for Plasmodium falciparum as well. So the CSP protein has been the candidate vaccine, as you may know, and the immunodominant part of the tandem repeat of that protein is the base for the vaccine of the RTSS or also the R21, which are both WHO approved vaccine currently in the field. So working with Plasmodium uelei, that let us develop a mice model where if we use a non-lethal strain of the parasite when we do challenge assay, we can look at the development of the blood stage of the parasite in the mice, and we can see if the vaccine is efficacious post-immunization following a challenge with sporozoite. For the purpose of this paper, and with the advantage, I would say the advantage of the replicon RNA is that we can clone in much larger gene sequence that has been studied so far. And the other innovation that we have here in this study is that we clone in the full length of the CSP protein. I
Okay.
just mentioned that our TSS or R21 candidate vaccine right now are based on the immunodomain, the tandem repeat of the CS protein. It's not the full length. Here we work with the full length protein. So you may think in terms of immunodominant anti-epitope, it has those, but it has also other epitope that may not be involved in the other vaccine. There's also the CSP protein has also a CD8 T cell epitope toward the C terminal of the sequence. So this is in. And also what we're thinking is it's important in terms of, the conformation of those epitopes. So I think having the full-length protein may have also something, may help in terms of the confirmation of presenting certain epitope
Mhm.
versus only a truncated portion of the protein.
And how different is the Plasmodium yolei circumsporozoite protein from that of Plasmodium falciparum? Because I noticed in your control you'd be using Plasmodium
Right,
vivax and falciparum
right.
control. So how different are they, or how similar are they?
Um, there is a I forgot exactly the percentage of identity,
Oh,
but there
we
is
don't
some
need the detail.
similarity, right?
Okay. Yeah.
But there is different from each other for sure. But we can have a little bit of cross reactivities
Okay.
sometime on certain ELISA assay that we have. So there is a few regions that may be a little bit conserved, but they may not be the immunodominant region.
Okay. Okay. You also mentioned that this parasite is non-lethal. So
So
does that mean uh,
there's two
is that normal
strains.
for this? Is there two strains?
Right,
Okay.
right. For the rodent malaria parasite, the P. yolei 17. There's two strains, a lethal strain and a non-lethal. So the non-lethal, basically, the when you do challenge in mice, the mice may develop parasitemia, but also may control the infection within 20 days, and it will resorb. The parasitemia will go up and go down. We had
Ah,
certain cases, case by case, we're following the mice day after day. Some of them develop really high parasitemia, so we have to euthanize those. We cannot continue or wait for them to resorb the infection by themselves.
mhm,
But at least with this strain, we can look at the blood stage post the challenge and see if the parasitemia is controlled. If they're first, we're talking about sterile protection. That means no blood stage appearance. If there's no sterile protection, if there is a development of a parasitemia, is there any delay in terms of the patency of the blood stage? So any delay in mice may be beneficial if you think about the term of development of the parasite linked with the symptom of the disease, right? So if there's a delay, maybe that could tell that there's a certain immunity, perhaps to help with having less severity of the symptom over time. But that obviously in the mice we can't tell. But I'm just saying we have different factors that we can look into the bloodstream. Um,
thank you. Dixon, did you have any questions that you want to ask
I
before
do. I have lots of questions, but I'm waiting
before
for
we
the
start talking
answers.
about
No, no, no. I'm waiting for the
okay, you're
answers.
waiting for oh, well, let's go through the paper then.
And
And
then
let
I'll,
me just double check. I am not sure if this is oh, it is open access. So
yes.
yes.
if our readers would like to read the paper or maybe would like to follow the discussion while we're talking about the paper, now would be a time to pause and click on the link that we give you in the show notes, and you can open the paper and you can look at the figures while
Right.
Marian is telling us a little bit more.
So, like I said, it was a rodent parasite that we're study because that's the mice model. So we clone into the P. yolei CSP protein full length. And as a negative control, we also clone the P. falciparum and the P. vivax CSP protein. And we know those will not have any effect in terms of protection. So the mice can still develop antibody, and we show that a certain respect on the paper. But those antibody has nothing to do with the protection against a challenge against Plasmodium yolei in the mice model. So we use those as an irrelevant repRNA strain instead of have an empty vector. We wanted to have the vector was another full-length protein. So we use a corresponding protein from a different species. So the self-amplifying RNA encoding the full-length P. yolei CSP were defined as our priming dose for our vaccine. And we combine that with the lion, like I said, the lipid inorganic particle developed by HD2 Bio. And one we look at first on figure number 2 was basically what happened with the prime and a boost regimen. Do we have good antibody, and do those antibody are protective against challenge? So we did a normal 2 weeks prime and boost regimen of immunization, and then we challenge with intravenous injection of sporozoite. So we got live sporozoite dissected from salivary gland from Seattle Children Insectarium for our challenges. So we inject them within an hour of being dissected, within an hour or two of being dissected, and you inject that intravenously in the mice, and those will create the challenge for your study. And then following the challenge, as you may know, and maybe I'll give a quick background on the life cycle of the parasite. So if you, in the mice is this basically same, except the timing is a bit different than human. But if you have an injection of sporozoite on our mice, we do an intravenous injection. But in human, obviously, if you bite by the mosquito, you will inject the sporozoite under your skin, and within a few hours, the sporozoite will go to your liver, which is going to be the asymptomatic phase of the disease. And then human is about 7 to 9 days, and then before it bursts out into the bloodstream, where it's become the symptomatic part of the disease. In the mice model, the liver stage is only 2 to 3 days. It's very quick, but the sporozoite will also, because we inject it intravenously, will directly go to the liver. It will establish himself in the liver, grow, and then if you have a naive mice, within 3 days, you start to see patency. So you start to see blood stage emerging in the blood in circulation in the mice. So what we've done is post-challenge, we immunize our mice, and post-challenge, we're getting blood smear every day, and we're looking at smear by eyes and quantifying the parasitemia. And we have naive mice for positive control, and we have mice we've been immunized with either the falciparum or the vivax CSP irrelevant RNA has a control group as well. So they all get the same dosage. It's 5 microgram for a prime and 5 microgram for a boost. And then post-challenge, what we saw, there were no protection at all. All the mice become positive about the same time. There's no delay in terms of patency, nothing. So prime and a boost, just an antibody-based vaccine, which we can show the antibody level here are really high, and the immunity is post-boost, post-prime, post-boost is there, and it's recalled post-challenge. It's there, really high quality antibody, really high titer. However, those antibody are not part of protecting mice against a challenge when it's just a repRNA only vaccine. So you may have heard in malaria, there's another approach of vaccine, and this is Dr. Sean's Murphy group at University of Washington that has been studying a prime and a trap vaccine approach. He's been using priming in his lab, is being interesting with a gene gun DNA vaccine approach for priming, like a subunit part of a vaccine. And the trapping dose, we call the trapping dose because we're going to use, instead of a subunit part of a vaccine, we're using now a whole organism vaccine. And the reason why we're using whole organism is because it has been shown that protection is linked with CD8 T cells, and mostly the CD8 T cell, liver resident CD8 T cells. And how to achieve those? If you use whole organism, so in that case, we're going to talk about an entire sporozoite, but you cannot inject a sporozoite live. You have to inject a sporozoite has been attenuated. Otherwise, obviously, you won't do the work, and you won't get the trapping dose to the liver to do the CD8 T cells. So a lot of the attenuated sporozoite are done through radiation, gamma radiation. There's also genetically attenuated parasite that you could create. So there is other group working on that, like the Stefan Kappe lab at Seattle Children's Hospital, been working on genetically attenuated parasite for has a whole organism vaccine approach as well. So for the paper here, we use radiation attenuated parasite. So we call that RAS, radiation
RAS.
attenuated sporozoite.
I have a wee question about those sporozoites because I'm just kind of trying to imagine what their surface coat looks like. Now,
Right.
if you think about trypanosomes, they have a very, very, very dense surface coat. So I
Yeah.
was just wondering, how dense is the circumsporozoite protein on those sporozoites, and what other stuff is on that surface?
Yeah. There's a lot.
Just to kind of, yeah, is there a lot? Okay, and is
A
it
lot
clearly
of other
visible as well?
things.
Right.
Yes,
Yeah.
absolutely. And
Okay.
CSP is one of the predominant, but there's
Okay.
other antigen, or another one called TRAP, actually. Not to confuse with the trapping dose, but yes, there's other antigen on the surface of the CSP, but the CSP is predominant, and this has been shown to be really important for the invasion of the sporozoite to the hepatocyte in the liver and the growth. So I should back up just for a second. The sporozoite not just invade one hepatocyte. The sporozoite will traverse some hepatocyte before taking residency in one specific hepatocyte. And the CSP antigen has been shown to be critical in all those different steps for the sporozoite itself. So the CSP is really one of the main candidates nowadays in malaria vaccine for targeting the parasite. So to come back for the prime and trap, so the priming dose, we thought it seems like encouraging to use a replicon RNA for a priming dose. We have a lot of great antibody that are, produced post even a single dose, but obviously it's not sufficient for protection. So we said, okay, what if we were combining with a trapping dose? So we decided to use the irradiated sporozoites. So same thing, we were getting freshly dissected sporozoite from salivary gland that we irradiated ourselves before injecting in mice. And that is what we have in figure, this is figure 4. Yes, in figure 4. I'll come back to figure 3 in a second. So figure 4, we started to look at a prime and a trap vaccine regimen, and we used the 14 days time of immunization versus a 5 days regimen of immunization. We were wondering if that would make any difference. So because we were doing the trapping dose, we were interested in figure 4 to obviously look at the CD8 T cells. Do that trapping dose still do the thing about the CD8 T cells? So this is a collaboration with Dr. Murphy's group at University of Washington. And what they have shown that I'll come back to the different regimen of immunization. But in general, what we've shown is, yes, the prime and trap was generating CD8 T cells, liver resident CD8 T cells. And we're looking at the KRLRG1 low and CR6 high memory T cells in the liver. And we show that we have produced the CD8 T cells of interest. The different color correspond to different dosage of the priming dose versus a different timing of the immunization. So like I said, the 14 days was here depicted in red, and then we went down to 5-day regimen here depicted in green. And we were surprised to see that actually the 5-day regimen we're actually doing better number of CD8 T cells if we're shortening the window between the prime and the trap than having the 14 days our regular priming dose.
And do you know why that might be?
Very good question. We do not know exactly, but we have some suspicion, but I'll come back in a second.
Okay.
But we don't have all the answer for that, I should say too. So obviously we're always controlling, with the irrelevant RNA group and also the trapping dose alone. So we also wanted to compare, obviously, a prime and trap vaccine approach again, a trap approach alone. So trapping dose would be just the irradiated sporozoite. Here we did a single dose of single irradiated sporozoite. We show in dark blue that we do have production of CD8 T cells. And you will see later they are responsible for a certain sterile protection. Just to back up as a general knowledge, the irradiated sporozoite has been in humans as well. You may recall some of the study by multi-group, including Scenaria, who's been producing
Vast numbers
sterile.
of sporozoites.
Yes,
Yeah.
sterile to be able to inject in human. And they do have clinical trial with at least, I believe it's 3 doses of irradiated sporozoite. And they need something like 900,000, I think, per doses to be injected to give some sterile protection. But you need the 3 doses and the 4th one probably a year later. So that's a lot of doses. That's a lot of sporozoite to be produced sterile. This is great, in terms of showing protection, but if you think about mass vaccination, is going to be a very difficult things to produce, right? So many sporozoite irradiated for mass vaccination. And
And can I ask, see, previously with the with the prime and boost, you got really, really high antibody
right,
titers, but no
right,
protection.
yep.
And now with this, you get you got T cell, you get a T cell response essentially. Um, did you also look at the antibodies?
Yes,
Yeah.
absolutely. So if we come back to figure 3,
Ah,
this
there
is
we
where
are.
we
Yeah.
look at the antibody. So we look at the different, again, the color code is the same. The red will be the 14-day immunization, and the green will be the 5-day immunization. The dark blue is the single irradiated sporozoite, always the irrelevant RNA first. But basically, it's just a single RAS point. So what we saw was very interesting, and we have part of to answer your question just before, that was what we show that is if we were waiting 5 days or longer to give the RAS dose post our priming dose, we were having a lot of higher, much more higher titer antibody. You can see that in the figure B. But also, that means we have less RAS parasite making it to the liver.
Hmm.
And in fact, what happened, all those antibodies that were produced post, in the second week of the post the immunization, were actually capturing our irradiated sporozoite upon injection. And they were not making it to the liver to do the T cell immunity we wanted. But so the liver burden here, the delta CT, might be not intuitive, but higher is the delta CT, less parasite you have in the liver. So
Okay.
we saw the red color dotted show when we have 14 days versus the green one where you have only 5 days versus the dark blue. If you just give RAS, if there's no antibody circulating before, they all go to the liver and you have a nice load of parasite in the liver and they will
Mm.
induce the CD8 T cells. But if we do more than 5 days regimen prime and more than 5-day regimen between the prime and the trap, we have such a good antibody titer post the self-amplifying RNA vaccine that they're actually capturing our irradiated spores that we inject in for the
Mm.
trapping dose. So
So you're
unfortunately,
essentially eliminating
yep,
the
yep.
trap vaccine section. Yeah.
Yes.
Okay.
So it was actually a pretty interesting data. We
Yeah.
we
Yeah.
didn't know that we were not expecting to get something like that. So when we saw that 5-day, we're like, wait a second. would it be nice if we can do it on the same day? Like basically, if you can do 5 days, but if you can reduce to a single clinic vaccine, right, instead of have the person come back within a week to get a second dose, would it be even better? I mean, if we get the
Yeah,
same antibody
absolutely.
titer and the same T cells, but can we do a single day clinic vaccine for the patient? So this is where the story went on, and we continue to look at figure, I guess, yeah. I'll do figure 5, sorry, before I show the same day, just to show you the efficacy basically of the prime and trap 14 days versus 5-day. So we were getting about 75 to 60% protection post challenge. On the 14 days, it was 60% in red versus 75% in 5-day immunization or regimen. And somehow expectedly, when we re-challenge all those mice later on, they were all protected. But I would say it's normal because they've seen the parasite once already, so they develop immunity against the first challenge. So that was not a full surprise, the re-challenge. But what was very interesting is, even though not all the mice were sterile protected, so that means no blood stages development, what we saw is like we have a consistent 2 to 3 day delay in terms of blood stage appearance in the immunized mice for those who didn't get sterile. And their parasitemia was much lower. They were able
Yep.
to control the parasitemia in addition of having a delay. So obviously, mice are not human. We do not know yet if it will be transposable, but that was very encouraging, because if you say, okay, I can protect maybe 70 or 80% sterile, but the 20% left, they may be able to control the infection perhaps better than if they were not immunized. That was very encouraging. So those data
Hmm.
were very good for us, because we're like, wow, we did a single priming dose, a single irradiated dose of sporozoite, and then we got already 70 to 80% sterile protection, which is what the WHO wants. They want 75%, protection. But this is, we're talking
Hmm.
about sterile protection, very stringent, protection. We want no blood, no patency. So to come back, following that, we said, okay, what happened if we reduce that 5-day interval to same day? Because I think that was really a thing that was very interesting to us to look for. And I should say, some of the people were not necessarily believing that we could achieve that. And that's basically the figure 7. I'm skipping figure 6, but figure 7, it's the same day versus 5-day immunization. In figure A and B and C, that's my study where we compare 5-day versus same day versus a like irradiated sporozoite as well alone. What we show, we have up to, on the same day, we had 1 mice out of 9 who were not protected, but her parasitemia was very low on day 12. There's still control. And the 5-day regimen still give again 80% sterile protection at 3 weeks challenge. Versus the trapping dose alone, the irradiated sporozoite alone, we're giving only 40%. So by combining on the same day a priming dose with irradiated sporozoite on the one dose was showing a much better sterile protection. And then the bottom part of the figure is we pushed the challenge way further. We went to same day clean, like a single immunization with a prime and a trap in mice. And we look at 2 months later with a higher dose of sporozoite, but they were from a cryopreserved sporozoite vial. But we pushed the the challenge 2 months later from that single immunization, single day visit immunization for the mice. And we still saw about 70% still protection. And again, a control of the parasitemia for those who become patent and a delay of the parasitemia. You can see on the graph of the when we plot the parasitemia per day, again, when we start to read parasitemia on the smear, it's at least 2 to 3 days beyond the positive control of the naive group. So it's quite promising. We're very excited to see that. We think there is a high potential here for our prime and trap vaccine, for multiple things. First, the replicon RNA with lion is fairly safe. It could be actually lyophilized. It's not here in the paper, but we've been working on and looking into how to make that dose, much affordable, costly first. And then if we can also reduce the number of sporozoite injected per person to, instead of 3-4 doses, to a single one, also the cost of goods for a vaccine in human could be much less. So yeah, this is about what the paper we published so far. Like I said, we've been working on an approach of looking into lyophilization, and we know it's capable of lyophilization. We have some data that has been published yet. We know the platform of replicon RNA / lion, not with malaria antigen, but with COVID antigen from our collaborator at HDT Bio, has been in many human. It has been in multiple clinical trials, is also in emergency authorization in India, and it has
Okay.
been in many human, and it's a lyophilized product. So we're highly confident that a priming dose, if we're going to move that to clinic in human, could be done even with a malaria antigen and involved in that priming dose, could be also a lyophilized product to reduce the logistic, the cost of goods, and all that stuff. And having a single day clinic visit to produce to help you having that immunity against malaria will be tremendous.
And that, I mean, this is really an exciting paper. I
Thank
really
you.
enjoyed reading that. Oh, I have to apologize. That was just a crazy dog running past me there, distracting me. I hope you can't hear him going wild in the garden. Um, So, actually, one of the questions that I did write down was whether that lion particle had already been trialed in humans, and you've
Yes.
just answered that to me, so that's good. And I think something that you said in the paper earlier on is an advantage of using lion would be that you could produce the lion particle and the replicon separately,
Yeah.
and then mixing it together. So I wasn't 100% sure what the advantage of that would be clinically. So I was wondering if you
Right.
could explain that to me.
Absolutely. For the paper, it's how it has been designed. We were producing the repRNA on its own and the lion, and we mix it basically at bedside of the
Okay,
mice.
yeah.
But when I say lyophilize, I'm talking about mixing them together and lyophilize the product combined already and reconstituted this before vaccination. So that we have now data that we did mix them, lyophilize, and put it in mice, and the immunogenicity did not change it at all.
Okay, so that's lyophilized the lion particle together with the
Right.
replicon
Yes.
RNA already.
Right.
Okay, that's
Yes.
good because that would make it a lot more.
Yep.
Well, I suppose you would be able to avoid the cold chain
Absolutely.
issues
Absolutely.
if you can just kind of reconstitute it at the
Yes.
bedside.
Right.
Ah, fantastic. Um, Dixon, do you have any questions? I
No,
still have I still have quite a few. I particularly wanted to home in on the sporozoites, but I don't know.
I
Want to
wanted
give
to
you a
point.
chance to say something.
No, I don't need a chance to say something.
Okay.
I'm fine, but I will anyway. I've worked next to people who were engaged in malaria research most of my life, and I know that it's not easy work, no matter
Mm-mm.
how you try. I go right back to the days of Jerry Vanderberg, who you might know that name because he was the person who radiated the
Right.
Plasmodium eualei, and he never got full credit for it. It was other people in the laboratory that took it over and ran with it. And,
Hmm.
there was a Nobel fever, I call it Nobel fever,
I see.
among the nuisance spikes, and they obviously got over it because they didn't get the Nobel Prize. At any rate, the point is that they didn't realize that the entire molecule was necessary
Hmm.
in order to induce full protection.
Right.
But I also know the work that Steve Hoffman is doing, where
Right.
he's working on producing in vitro
Absolutely.
sporozoites
Yep.
to take all of this out of the ballpark. Just give them the radiated sporozoites and you're done. So what do you think of that?
Oh, I think we we've been working, and we know Steve Hoffman group as well. They know
I'm sure.
we work on in vitro sporozoite as well production. We one of the two lab was his lab that can produce some sporozoite in vitro.
Yep.
That would be a game changer for sure in terms of, if we can produce those sporozoite in vitro, it does not have to dissect mosquito. That
Right.
would be definitely a game changer. The Oh, I just lost my thought. Oops. Um,
That happens to me all the time.
what was I going to say?
Dare I say it happens to me as well. So
Sorry. um, all right. Anyway, maybe that'll come back to me in a second.
Yeah, we were talking about in
Um,
vitro production of
right.
sporozoites.
Yes, no, I think producing an in vitro sporozoite has a whole vaccine, and for trapping those, for example, here, or even to replace those mosquito heavy, human involved dissecting million of sterile mosquito.
yeah.
It's just yeah, it will be great. We're also, I mean, we're working also on looking into maybe a synthetic trapping dose, to be honest, because even though we're very, very much happy with having just a single dose irradiating sporozoite, we would love to maybe work into maybe using a genetically attenuated parasite instead of irradiated sporozoite. As you
Hmm.
may know, you just need one sporozoite to make it through the liver and get to the
That's
bloodstream.
right.
So
Yep.
That's
I'm
right.
not saying there's no risk. There is risk to use a
Mm.
whole organism as a vaccine or trapping dose. But still, I mean, I think if we can, we at MalarVx right now, we're actually our plan is to push a prime and trap through a phase 1 clinical trial. Actually, we
In
really,
people.
yes, to see what's happened in people, because again, like I said, mice are not people. So it'll
Some of
be
them are.
so they will be very, very, very informative to see. Just to back up, maybe for those who are listening and not necessarily familiar, but the replicon and the lion has been in million people already through with another antigen than malaria, but COVID antigen, for example, has been in multiple clinical trial and in million people in India. There's up to a phase 3 clinical trial data available with that if you look at HDT Bio. The irradiated sporozoite has been also in million people through all the work that Steve Hoffman and Seneria and his collaborator have done through immunization in clinical trial. So we know each of those components, and the CSP protein, the antigen of interest here, has been in million people because that's the protein, that base protein used in CSP and R21. So now we're taking all those little different pieces, we put them together, and we show there is a synergy, which still we don't know exactly how. We're still investigating this, but there's a synergy of doing the prime and the trap together, because there's something going on with the immune response here, and we haven't figured out all the little pieces here, but there's a synergy happening when you do that prime and trap injection together that works for the right immunity that show protection. And we believe this should be go test in human. It has never been done. And to be frank, the same day single clinic visit, a lot of people were like, I don't think it's going to work. But then when we're starting to show data, they were like, wow, we can talk about maybe mass vaccine campaign immunization if that do work, right? Because you ask the person to come once and you maybe have 2 different injections, because a repRNA right now it's an intramuscular injection, and the irradiated sporozoite is an intravenous injection. So it's 2 different routes of injection, right? 2 different products. But single day visit, it's tremendous for the people living in endemic area, make a big difference.
So do you need to inject intravenously those doses?
The
The
sporozoite,
sporozoite.
yes. Yeah.
Yeah. I suppose
The
that would be a
That's a
bit of a
problem.
barrier then. That
That's a
would be
problem, right?
problematic because
right.
you'd need to not just get
It's a
vaccinated, you'd get
problem.
medically trained.
Yep.
I suppose
Yeah,
nursing
well,
staff or doctors.
Yep. That is
Yeah.
still a little thing. Plus, you still have the chain code for that specific dose,
Yeah.
because sporozoite are cryopreserved.
right.
So yeah, you still have this, at least for one of the 2 doses, we still have that. But I mean, it's still some progress, we're
Oh
still
no, it's an
gonna
Of
amazing.
see.
course, of
It's
course, of course.
it's the proof of concept,
Yeah,
really. I think, and I think what's really nice with that approach is kind of current existing vaccines are just were based on the circumsporozoite
right.
protein, and they presumably, I can't remember. We did discuss the papers, but I can't actually remember the details of the immune response. But presumably, will be very highly shifted towards an antibody production.
Right.
Whereas with your approach, you're actually really
Both.
activating both arms of the
Yes.
immune system, which I
Yes.
think is really, really important for
Absolutely.
sterilizing immunity.
Yes, and you're right,
Yeah,
because all the RTSS and R21 are humoral immune response driven.
yeah.
It's antibody mostly. So yeah, you I think we do think there is a high potential here in the technology, and the approach, the innovation of the same day vaccine. There's not many vaccine study in general that try to do that prime and boost or prime and trap 2 doses on the same day. We always think, oh, we have to space, and we have to space to give the immune response. Actually, for us, I should say, it has not been published, but if we do a prime and boost, the repRNA lion is so potent in the mice. We track the antibody up to more than 3 months later, and we still have antibody in circulation at really high titer. So regardless
Hmm.
of the delay we want to put, our trapping dose following the priming dose, we think if we give it on the same day, it boosts already boost. You already engage your T cells, and you are starting to engage your antibody, but then they are not annuling each other because the antibody is so potent. And I should mention, it's not, I haven't mentioned that, but also the lion lipid inorganic nanoparticle, it has shown a lot of more safe than most of the LNP that we know currently. So it's also something very positive here, if I may say it like that. So we're very, very fortunate to collaborate with HGT Bio in this priming dose approach of a repRNA lion. And yeah, it's been a tremendous collaboration in that. And we have taken on the malaria vaccine approach with them on that. And I would say it has been tremendous journey since we started with them. So very grateful for that. And like I said, all the people in that paper, they all have all an important role. And it was really a great pleasure to work with all of them.
It's just such an exciting, really, paper, such an exciting set of experiments. I had something else, but just like you earlier, it just slipped my mind again. I have the excuse of it being very late at night.
Maybe I can help you out.
You
How
help me out,
long
please.
after you initially immunize the mice can you infect them and still have protection? What's the longest you can wait before the protection wears off?
Um, Like I said, when we haven't tested that, but what we look into is, like I think mentioned, we did a prime and boost
Yep.
with the repRNA, and we still see the antibody 3 months later in
Well, the
circulation.
antibodies are okay, but
Right.
did you challenge the
Oh
mice?
no, not in that case. Not in that case. We do not. And
I
we
mean,
know,
mice only live 2 years, so you don't
right.
have too much time to wait before you hurry up and get them.
But we did not know. We did not want to challenge those because it was just a permanent boost, and we know just repRNA alone will not be protected. So in that particular case, we're just looking at the longevity of the antibody life, not at the efficacy of, because we know prime boost regimen will not protect. We need the prime and trap for protection.
Yeah.
What about irradiated sporozoites just by themselves?
So
How long can you wait afterwards and still have
yeah,
protection?
so if you do the challenge at 3 weeks, you have the 60% on average, 60%,
60.
60, yeah, 60% protection. But when we push down to 2 months challenge, and we did that in figure, in the last figure of the paper, figure 7, when you do 2 months challenge, you drop down to 25 to 30% maximum protection with just irradiated sporozoite. So the irradiated sporozoite, it wind off pretty quickly. You won't keep that protection around very much.
Yep. Yep. I realize that from
Yep.
the very everybody I've ever spoken to has they were amazed at how short-lived the immunity really is.
Right.
Because in a real situation, you can get malaria every year,
Yes.
and so that tells you something.
Right.
Now, what kind of T cells could they be to have such a short life?
Mm-hmm.
That's a big. And how can you make them live longer? That's the
Right.
idea.
That, and then, I mean, it's something, obviously it's very difficult in mice model, but I think the
Yeah,
pre-exposure part for me
that's
is
true.
an
That's true.
important point. And I mean, I can't really do that right now with mice model, but pre-exposure, I think, has also a lot of things, very interesting immunological question that would be informative to study, I think, in the primary.
Sure, sure, sure. The other question I had, which is kind of weird, is that do patients or people, or let's say mice, because you're using Anopheles stevensii for your vector, when they feed upon a mouse that has high titers of antibody,
Mm-hmm.
do they produce fewer sporozoites as a result? Even if the mice
Um,
have a live infection?
they may. Um, I don't remember that on the top of my head. They might. If they had the right antibody.
Right.
Um,
But
hmm. I
but you were not feeding the you were not feeding or infecting the
No,
mice with the mosquitoes, weren't you?
no,
No.
no, no, not at all.
No.
We
No.
were for the challenge, and this is something we, yeah, we haven't done because limitation. I mean, we're a small team at MalarVx, so unfortunately we're lacking manpower to do too many things. But what we were doing, all the challenge and everything was just done with freshly dissected sporozoite. So we
Right.
never had, uh, yeah, like like I said, like a mosquito challenge.
Okay.
I suppose you can control the dose of infection much
Right.
better if you do isolate your sporozoites and
Yes.
then,
Yes.
yeah, because otherwise you don't quite know.
It is a guess, yes.
Yeah,
You can
yeah,
control because you can, I believe, dissect the mosquito later on and look at how,
yeah.
you have some batches you dissect before and other after the feeding, and you can compare, blah, blah, blah. But yeah, we
But that
haven't
was
done
just
that.
an extra step of work.
Yeah, and we haven't done
Yeah,
that.
yeah,
In our
yeah.
lab, we don't have the insectarium, so we're depending on gracefully from the Seattle Children Insectarium here in Seattle. So we have to be very quick upon dissection to come back to our lab and inject our mice with. So yeah, I mean, there's so many. There's so many questions I would love to.
Sure
Well,
there.
but you've got your entire career ahead of you. So
Yeah.
next stop will be the clinical trial,
Yes.
maybe.
So absolutely.
Um,
We're
yeah,
gearing
and
this
then we'll have you
actually.
back.
Yeah. We're
Yeah,
gearing for that. We're doing right now, we're producing our own priming dose in CGMP manufacturing.
good,
Yeah.
good. And
So
then we'll have you back, and you
I
can
would
tell
love
us all
to.
about the success of that. So I thank you very much for telling us about your
Well,
paper.
thank you
I
for
really,
having
really
me.
Yeah.
enjoyed that.
Thank
Well,
you
it's
so
good. Yeah,
much.
it was a really interesting paper.
Yeah, thank you.
Yeah, so what shall we do next? I have a hero, so shall
You
I shall
do
I
have a hero.
Yeah,
Go,
I do
go
have
for it.
I have so many heroes. So today's hero is a woman, Mweli Ntuli Malecela, and I had to look up how to pronounce. I had to listen to quite a few YouTube videos, so I got the name pronunciation right. So I hope I have. So Welly Malichela, I would like to talk about her today. So she was the head of the WHO neglected tropical diseases section,
Right.
and she, at the end of her life, she led the neglected tropical diseases roadmap that we have talked about in a previous TWiP. I think it was maybe back in December, when Chuck Nurse was on the podcast as well. So Mweli Malecela, she was born in 1963, so she's not much older, or she was not much older than I actually. Um, she has achieved so much. So she was born in Dar es Salaam in Tanzania, and she grew up in a prominent political family. She was actually the daughter of the former Tanzanian Prime Minister and permanent representative to the United Nations, John Malichela. So she was educated in Dar es Salaam, and she went on to study biology and graduated in 1986 with a degree in zoology. And then, after her studies, she joined the Amani Center of the Tanzania National Institute for Medical Research, NIMR, where she started working on lymphatic filariasis. So she was particularly interested in Bancroftian filariasis back then. And then she went off to London, to the London School of Hygiene and Tropical Medicine, where she did an MSc, which she completed in 1990, and a PhD, which she graduated from in 1995. And the focus of her research all that time was filariasis. Actually, the title of her PhD thesis was The Immune Response and Microfilaria in Cats Infected with Brugia pahangi, so not a human parasite,
Did
but
you?
a very similar filarian, a very similar parasite to Brugia malayi. I think, Dixon. I don't know if you know much about. I don't really know all that much about Brugia pahangi, other than it infects cats, and that we
Right.
can have it in the lab because it's not
Well,
harmful for students.
I had a very good friend who was in charge of that program, by the way. His name was David Denham, and I'm guessing that she got her degree with David.
Yes, that's what she did.
I
That's
knew that.
absolutely
I
correct.
used to the joy of going to the London School of Hygiene and Tropical Medicine was that they had their own tavern in the cellar. So you could always, if you got a good result, you could go down and celebrate. If you
Oh,
got
so
a bad result, you could go down and moan.
and you
But
could
it
just
didn't
roll
matter
down the
because you
steps and
could
you
always
were
go
right
down and have
there.
a drink no matter
Perfect.
what. And so David worked on the cats, and he worked
Yeah.
on immunity, and I'm sure that's what she did her work on also.
Yeah. So we have in the past used Bruja Bahangi as a kind of a model just to show students
Yeah,
what they look
that's right.
like
That's right.
in culture. So it's
That's
quite
right.
interesting to watch that. Anyway, so when she did her PhD, she went back to Tanzania to NIMR, and she continued her research. And then in 1998, that was only like 3 years after completing her PhD, she became the director of research coordination and promotion. And I was kind of thinking about that timeline. So, completing her PhD in 1995 and then become a director in 1999. That's a very kind of quick promotion to senior position in a very short time. And I think that really illustrates just how talented she was. Anyway, in that role, she was instrumental in preparation of the first national lymphatic filariasis program for Tanzania. And then 2 years later, she became director of the lymphatic filariasis program. And in 2010, she was appointed the Director General of NIMR, and she was the first woman to have that position. So she did grow up in a political family. I mentioned that early. So she was politically active as well and a member of the ruling party in Tanzania. And she contested during the 2015 um, p- um, party presidential primaries as one of few women, because I suppose sh- she maybe wanted to kind of expand her influence more into the political policy kind of domain. She was, however, not successful, and she went back to being a scientist. Um, and she was just um, continuously focusing on filariasis in that time. She was dismissed in 2016 from her post as director over a controversy over Zika virus infections in Tanzania, because
Mm.
that was not really something that, the Zika virus infection was not welcome, essentially, but present. But very soon after, so less than half a year, she then joined the WHO in 2017 as the director in the office of the WHO regional office for Africa in Brazzaville, and she oversaw policy implementation there. And then a year later, everything goes really fast with her. She became director of the Department of Control of Neglected Tropical Diseases. And in this role, amongst other things, she established the Youth Against NTDs to improve involvement of young people in NTD programs. Um, so uh, in that time, so in that short time as a director, so she really, I think, it, the culmination of her work as a director culminated notably in the development of a new roadmap for the 2021 to 2030 roadmap, which was launched in 2021 and which we talked about on TWiP before. So she received lots of distinctions and prizes, and she was included in a list of 100 most influential women in Africa. And she also advocated for the empowerment of women and gender equality and the welfare of women and girls. Unfortunately, she was diagnosed with cancer in 2018, and she died 4 years later from cancer. So that was really sad, because of who knows where she would have gone if she had a little bit more time. And one of her legacies, though, that are still ongoing is the Mweli Malichela mentorship program for women in neglected tropical diseases, which is a program that supports mid-career African women to become leaders and champions in NTD elimination, both nationally and also internationally. And this is also aligned with the global NTD roadmap, and will provide mentorship and training and networking opportunities for women, um, over 2 years each. And I think that is a really amazing legacy to have. So I think, she is remembered in the community as a really inspirational figure and a dedicated leader. And I'm sad that she's no longer with us and can continue her amazing work. So that was my hero. Yeah, so I'm worn out.
Not easy being an MC, is it?
No, it is not. Now I've also lost my show notes. I'm going to
Oh
have to
no!
try and.
Oh
Have
no!
you got your show notes? Ah, there we are.
Just
Okay, so I suppose I don't know, unless we have any other pressing things that you would like to say, um, we can maybe think of wrapping up. Do you
sounds
want
like
to
a
do
good
the wrap
idea.
up, Dixon, or do you want me to do
Sound.
it?
Oh, would you? Well, I can do the end part. How about you do
Okay.
the rest?
Okay, I'll do the rest. So first of all, I want to thank Marion.
Absolutely.
Sorry, this is terrible, Marion,
There.
for coming along tonight and talk about your exciting work. I really enjoyed chatting to you and learning more about the work that you and your company do.
Pleasure to be here.
Um,
Thank you so much. I
so
enjoyed it.
good, good.
Good.
So that's TWiP 236. You can find the show notes at microbe.tv/TWiP. If you have questions or comments, you can send them to twip@microbe.tv. And if you like our work, you could consider supporting us at microbe.tv/contribute. Even just a coffee worth of donation would be amazing. So I've already thanked our guests, but I'm going to thank you again. So thank you, Marion Avril at MalarVx. You can be also found at Twitter. I think that's your Twitter handle at Marion Avril FR. So if you're on Twitter or X, you can look her up there and follow her. Dixon Despommier. I'm just really messing up my names.
Oh, you
Dixon
did it
Despommier.
perfect. It's
No, I
perfect.
did not. Dixon Despommier is at trichinella.org and thelivingriver.org. And also, I would like to push Dixon's book.
Oh, please
Um,
do.
yes, so go and have a look for Dixon's book. So thank you very much, Dixon.
You're welcome.
I'm Christina Naula. I'm at the University of Glasgow. We would like to thank the American Society for Microbiology for their support of TWiP and Ronald Jenkins for their music. You have been listening to This Week in Parasitism. Thanks for joining us. We'll be back in 2 weeks.
Another TWiP is parasitic.